Okay, so thanks everybody for coming to this Kirby seminar, both here in the room and online. And I'm very excited that we have Professor Ewa Goldys from engineering presenting today. I'd like to start by acknowledging the traditional owners of the land on which we meet. And in this room, we're on the lands of the Bijigal people. And I'd like to extend pay my respects to those people and extend that respect to elders past and present. So today we have a presentation from Ewa Goldys, who is, as I said, from engineering. And Ewa has a career of developing fluorescent and luminescent nanomaterials and using them for biological purposes. She has a storied career which is included leading the ARC Center for Excellence on Nanoscale Biophotonics. But Ewa has really an unparalleled track record of being able to work with industry while pursuing her academic pursuits. In 2016, she was awarded the Eureka Prize for her work in being able to look at naturally occurring biofluorescence and using that to fingerprint both healthy and diseased cells. So why I'm really excited to have Ewa here today is that a lot of you work on point of care testing and a lot of you work on new models of care. And we use very traditional technology based on PCR or isothermic amplification. And we use that to very good effect. And we're really learning how to use that more efficiently all the time and base not just diagnosis, but treatment algorithms off the back of that. But just imagine if instead of having a little machine that did that, you could do this on a handheld device that looked like a rat test. So Ewa is developing technology that may allow us to do that. And so I'm going to hand over to Ewa to tell us about how this might work in the future. Thanks very much.
Good afternoon, everybody. Thank you, Tony, for your kind introduction, and to Tanya as well for inviting me over to speak to you. I represent biomedical engineering. My exposure to biosciences has been over many years. I've been developing different technologies for chemical sensing, also on a nanoscale level. But since about 2018 or 2019, we became really interested in some new technology that has the potential of really transforming the way end user sensing is done. So I will tell you a little bit of that story in a moment. So I'd like to acknowledge my collaborators here and co-inventors of this technology. And this is my first slide. That is actually my proper introduction to PCR, which is polymerase chain reaction. As a physicist by training, I didn't know what it is unless I had to stand in those queues for a long, long time. And I then was told that this is really needed because we need to send the sample to the pathology laboratory. And that was not my first introduction to biosensing, but I already knew that this logistics brings with it a lot of costs, a lot of complications, and obviously costs were here to the health system. That made us think, what else, where else this DNA and RNA testing technologies could be needed? And Tanya told me that I need to present a context of the technology that we are developing. So I have a few slides which I will share with you. So clearly we are in Kirby, we are talking infectious diseases, completely not my area. So I'm hoping that at the end of this seminar, we are going to have a discussion where you will tell me exactly why point of care diagnostics is really needed in infectious disease. This is a few of different bullet points. Rapid testing in clinics, pharmacies, remote settings. Obviously, pandemic preparedness. We need quick deployable tests in the case if there is some new disease lurking and trying to explode. Veterinary diagnostics, those of you who have dogs would know how extremely difficult it is to diagnose whatever is wrong with the dog. And having end user test strip for this would be really, really super important. And then there are more complex things that have to do with monitoring treatment responses in the medical setting. Now, this is an interesting area. I became aware of environmental DNA testing a few years ago. And since then, we sort of initiated a program on looking for environmental DNA. So at the center of environmental DNA testing is the concept that DNA is a very resilient molecule. It is present in cells, one piece of DNA in every cell. It is species specific, and it can persist in the environment sometimes for decades and maybe even longer. So having, as the different organisms exist in the environment, they would leave behind the traces of their DNA. And those traces are able to be detected. They are currently detectable in the area of environmental sciences, and they can be utilized for a whole lot of really interesting things. You can detect invasive species with it. You can also look for endangered species in a way that doesn't stress the animals. It is possible to sample water or sample soil for other things, and many of you would have heard about concept of sewer analytics. So that is actually has been in the media during COVID period, testing of sewage for traces of in this case, COVID, or other diseases, like whatever they are testing for different things like illicit drugs or antibiotics or things like that. Obviously, identifying harmful microorganisms in water is very important. Also, from the point of view of water industry, they need to make sure that the organisms that are normally present but harmful can be suppressed or eliminated. One can look for traces of contamination, heavy metal resistance genes in microbes. This is one of the few different applications where end-user friendly nucleic acid tests would be really, really valuable. All of those applications exist currently, but they are done using PCR and similar techniques. So it's not as widespread as it might have been. There is high cost to it and limiting the usefulness. There is a whole lot of medical uses of nucleic acid tests beyond the infectious diseases. I've got a few here and some other different bits and pieces of applications, but I would like to tell you a little bit of a story of cancer-related mutations. Most of you would know that cancer is associated with mutations in the DNA. Some mutations are known to be leading to increased susceptibility of individuals to cancer, but some mutations are actually present in cancer. A very interesting, they were discovered in biopsies from cancer. And obviously, if a patient is undergoing surgery, there is a sample taken, and that sample will be tested for the presence of mutations. However, that is a one-off opportunity, because generally speaking, patients do not get repeated biopsies. However, it has been discovered that cancer cells, they released nucleic acids, they release DNA into the bloodstream. And that DNA keeps circulating because, as I mentioned, it is a resilient molecule. It's not going to degrade very easily. And it is sort of keeps walking around in blood. It's present at very low concentrations, however. So it's quite difficult to convert from the medical literature to engineering literature, but we spend a lot of time working this out. It is between 1 and 100 copies per microliter of blood plasma. And so it is a really challenging detection region. It is the region of PCR. And generally speaking, the cancer mutations are detected by methodologies similar to that. And obviously there is cost, there is the lack of problems with repeated sampling. Sometimes they need a lot of blood because blood needs to be enriched in those nucleic acids. And furthermore, the nucleic acid in the blood is mostly coming from immune cells. But the tumor mutations might be present in the tumor, but not in general body. So on top of rarity of those cells, we have a problem that only a small fraction of those cells have mutations. So really major challenge for the detection. However, the payoff of having a convenient and low-cost method for detection of those mutations would be great, simply because the cancer, as it is being treated, is undergoing evolution and transforms itself. And it happens sometimes that those worse subpopulations in the tumor start to dominate, and or maybe not, if the treatment has been effective. So looking at the evolution of what those mutations are doing during treatment, that is a very significant objective for a lot of clinical medicine. And as I said, we have existing technology, but it's NGS technology. It needs the sample to be taken to a scientific laboratory, not even to pathology. So we need something better. I have here a number of different other applications, so I'm not going to go through them in detail, but I just want to sort of flash them in front of you here. Food industry, very important technology. Obviously, you would want your sausage to be completely safe to eat. Now, many products in the food industry are fermented, so there is microbial fermentation going on, and it's all good if it is going the right way, but you can have wrong species being bred. What is very important for the food industry is to be able to finish the production of that sausage during single shift. And what if the sample needs to be taken to a pathology laboratory, and the pathology laboratory is in Melbourne? So I had conversations with a whole lot of industries here to try to find a way the contamination can be identified in a single shift. They have methodologies, but they use culture. So it's all sorts of challenges. Livestock and aquaculture health. You heard about salmons from Tasmania being killed in thousands and thousands. So being able to detect this kind of disease outbreaks in fisheries would be really important. Biosecurity monitoring. We had discussions about biosecurity recently. And so this is how I learned some weird names like sea vomit, which apparently is an algae that grows on ships, which is invasive, and we don't want to have it here in Australia. And it's very, very difficult to detect. You have to put sand divers. So all sorts of different applications for end-user-friendly nucleic acid tests. Okay, so this is the general context. ChatGPT doesn't know anything more. I told you my stories, but this was ChatGPT. So now I will tell you about a new way how nucleic acids can be recognized. And I am very excited to discuss this because, in my opinion, the area of biosensing undergoes a transformation which is very similar to what happened about probably 40 to 50 years ago when antibodies first came to the scene. And antibodies have given us technologies such as ELISA, which is now commonly used everywhere, which are able to detect different proteins with great deal of sensitivity, used in medicine, used in biology, across a whole range of things. Now, antibodies is the response of higher organisms to infection. Now, about probably 10 years ago, there was a great advance in the understanding how microbial species deal with infection and infection by viruses or phages. And that, obviously, you all know, this has given rise to a Nobel Prize in 2020 for the discovery that Cas9 protein can be used for gene editing. But in addition to application of these proteins responsible for microbial immunity to gene editing, there was a growing emergence of a smaller story, a story how they can use in biosensing. So I will tell you a little bit more about those proteins and how they can be harnessed to detect nucleic acids at a high sensitivity. So this is going to be a discussion about mechanisms. So I will tell you about proteins. These are the proteins. shown here, and the colorful objects here, the blue ribbons and the yellow ribbons, are nucleic acids. Now, it is important to count the actors. So I will be talking about a protein, mostly Cas12a, which looks like this, the gray ribbons. And there will be 3 types of nucleic acids mentioned. I will talk about guide RNA. Guide RNA, which is this yellow ribbon here, it conjugates to the protein and it sort of stays there. I will also talk about 2 types of DNA molecules. One type, which is called the target. This is the pathogen DNA that we'll be detecting. And I will also talk about other small pieces of DNA, which are the engineered nucleic acids, which will be cleaved by Cas proteins, which are nucleases, and we call them reporters. What is so very unique about those proteins is that the proteins can be programmed. And the program is available in this sequence. The guide DNA contains a sequence, nucleic acid sequence, that contains the program. And what this program does is this. At the beginning, where we do not have a target, the protein integrates with the guide and pretty much does nothing. It is not yet a nuclease. It will become a nuclease in a moment, but for now, it just sits there, conjugated. The moment a nucleic acid target comes to the scene, and if it has a sequence that is precisely matching the guide sequence, then this converts the protein into an active nuclease. And that conversion is through conformational change. So you can see here probably that there is some degree of shape change. And basically it's like claws that open, and when they become open, the nuclease starts to cut. So this is the symbol that I will be using. This is the inactive protein, and this is a programmed protein that acquired its blue target and therefore changed into a nuclease. So we call it activated. Now it is ready to cleave. Now, the cleavage is of 2 different types. One is the cleavage which only cuts the DNA sequences that match guide RNA. Those of you who are familiar with gene editing, they would know this is how gene editing works. But I will be talking exclusively about transcleavage, which is the non-specific cutting of any structure, any sequence that comes here. And this is the one that is useful for biosensing. So now I will tell you historical developments. This is how a basic nucleic acid sensor works, CRISPR sensor works, and then I will tell you about the modification that we have made. So this is the literature, how a sensor can make. I have to say that the moment I heard it, I thought, geez, this is brilliant. How is it possible? So we bring together Cas12a ribonucleoproteins from, I just told you that they have an integrated guide. They are already programmed. And here in a sample comes target nucleic acid, right? So we are putting a few of those proteins with a bunch of targets, and normally more proteins than targets because, you know, the pathogens are not that abundant. So some of those proteins become active. They are able to cleave nucleic acid. The rest doesn't do anything, so we ignore it. The next stage, we add to this mixture quenched fluorescent reporters. These are pieces of nucleic acids. Here is nucleic acid, and it has 2 molecules, a fluorophore at the end, fluorescent molecule, so the same technology that is used in our lamps over our head. And on the other side, there is a molecule which is called quencher, which suppresses fluorescence from that reporter. So I intentionally use this dull red color because this whole moiety is not doing much. There is a tiny bit of fluorescent light, but really just basically very little. Okay, and at the next stage, what happens is that nuclease kicks in, starts cleaving those reporters, and the moment it cleaves, the quencher, which is killing the fluorescence signal, disappears, goes away. I didn't even draw them because they are invisible. And the rest, the only molecules that are visible are those fluorescent moieties that are now on their own, so able to produce light. So this is how we end up with fluorescent signal. Now just wrapping up, this is how testing works. The sample with targets is brought together with Cas12a ribonucleoproteins or RNPs. Then this happens, and then reporters are added. They can be added earlier as well. After minutes, we can read the signal. Great technology because it is a single pot technology, but it has a whole lot of good features, high specificity. We can detect nucleic acid in a sequence-specific way. It's pretty fast, 5 minutes, 10 minutes. Lovely feature, isothermal detection. You can do it at room temperature. And even if the temperature varies, it's still not a problem. Compatible with downstream methods, single pot, no wash, fantastic. You can actually make test strips with it, which is called lateral flow assays. Test strips look like rat tests. And that's precisely what they are. There is a slight problem. If you do not do anything to this design, the limit of detection is picomolar. So that is not enough for many clinical applications, and especially for infectious diseases. Now, if there is a really bad infection, like fully developed COVID, then there is enough sensitivity. But early COVID, low-level infectivity, that's not enough. So most researchers would include pre-amplification step, such as PCR or LAMP, but now we are back to square one, because PCR needs equipment and it needs temperature cycling. LAMP, amazing, isothermal, but high temperature. It's 65 degrees, if you are lucky. So that is a problem, and we tried to solve this problem. And in order to solve this problem, we introduced an additional component. But before I start to describe this component, I would like to just draw this piece of schematics to just summarize what I have shown you previously. So a standard CRISPR sensor has this programmed RNP and a target, and then when they come together and integrate, the Cas12 protein becomes a nuclease, starts cleaving. And what is cleaving? It is cleaving the reporters here in an enzymatic loop. It goes around in circles and just keeps cleaving, 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 cleaving. So that's an enzymatic loop, okay, catalytic loop. What we have done, and this is the innovation that with my colleagues have been able to bring to the field, is that we have actually done something else, which is well illustrated in this slide. This slide shows dancing dervishes. So they swirled around their own axes, but you probably have seen those movies, right? They also go around in another circle. So there are two circular movements going on at the same time, and that's what we have done. So you should imagine that the small enzymatic loop of cleaving the reporters, it's the movement around the axis, and we introduce this big loop. We make everything move in a big circle. And that has been achieved with the addition of a special component. This is an engineered nucleic acid structure, which is in the form of a circle. In the publication here, Nature Communications, it was called cell mediator, but now we just use nanocircles. It is a super small structure, about 2 nanometers in diameter. And overall, this is a single nucleotide here. I counted them. This is exactly how it looks like. There are 3 nucleotides here that are single-stranded, and the rest is double-stranded. Once of the part of the sequence is special, it is called a PAM sequence. It is TTV sequence, which is needed by the protein. Now, the purpose of those different pieces is different. This is the place where we are provoking the protein to cleave. And it will do that because it preferentially cleaves single-stranded nucleic acid compared to the double-stranded. And this region here is around 16 nucleotides. It can be slightly longer sometimes. This is the structure that is long enough to activate the protein. And what happens is this. This is this big autocatalysis loop that I have foreshadowed. I will take you through this, starting from this point here. Let's imagine that by some methodology, which I will show you in a moment, one of the proteins have found a target and became activated. Okay, so now it is activated. Now we are giving that protein the circle, the SER mediator. It's still closed, but the protein recognizes it and it's now ready to cleave. And what is it going to do? Well, there is a provocation here. Let's cleave here, right? So this circle is going to become cleaved, then it linearizes, but its sequence is matching the programmable, the guide in that protein. So after linearization, this circular structure becomes a fake target. We called it simulation target, but it's a fake target. That fake target is present in the mixture, and in the reaction mixture, we have also inactive RNPs. They have the guide for that, and now they recognize, oh, here is that structure that matches my guide. And a new protein comes in and becomes activated. So we now completed the loop. I started from one activated RNP that appeared by some magic, but now I have added a circular mediator and inactive protein, and now I have two active proteins. So this is the methodology that breeds active proteins through fake targets. Okay, so the entire scheme looks like this. I told you that the first activated protein needs to come from some magic. It doesn't. It comes from my original CRISPR sensor. So this is an example of a scheme where we have two proteins programmed for two different guides. This is the pathogen guide. I can't choose it. I mean, you are telling me what is going to be the pathogen. The DNA is very long. I only need about 20 mer out of this long sequence, but it is still pathogen specific. Where I will find it, I will go over the genome. I will try to find where is the PAM. And then I will take a gamble, and that gamble normally works, sometimes better than in some other cases. So this is this first CRISPR sensor. The protein gets activated, keeps cleaving the reporters. We don't mind it, let it cleave. However, this protein is also able to have a go at my circles because transcleavage is agnostic with respect to the sequence. And so now I have this circle, has been cleaved, becomes a linear fake target. It finds the blue proteins, which I also provided to the reaction. This is then going to become active. The moment it becomes active, it can have a go at my reporters. So it keeps cleaving, but it can also cleave that circle. It has a choice. And so these are the small loops. These are the dervishes swirling around, and this is the big loop, and everything keeps swirling and swirling. A lot of swirling. And so what this does, it really transforms a CRISPR sensor, which is already pretty good, picomolar sensitivity, into a complete explosion. Yesterday, I calculated the amplification factor. Now, if everything cleaves nicely, the factor is, I just don't even want to tell you the number. It's 10 to the 35 in 10 minutes. It's unrealistically high. It is crazy. So in a conventional CRISPR sensor, we have a one-to-one correspondence between the targets and activated proteins, because that's how it works. It needs to integrate. And then the catalytic loop is going to produce quite a number of fluorescent reporters. And that gives rise to the final fluorescent signal that is already pretty good. But here, this is what is happening. One target DNA produces one activated pink protein, the pathogen matching protein. And then we have a whole lot of proteins that are activated by fake targets. And each of them is cleaving that much, and so you get this explosion. So this is why this is an advance, because we broke the one-to-one correspondence, and now we have one target activating a whole lot of proteins. We calculated in the published paper, we calculated how many we are obtaining in 10 minutes, which is a pretty high number. And this is what brings the amplification to the system. But this is not an amplification through amplicons. There is no amplicons here. This is just circles being converted into fake targets. So it is a capped amount. Now contamination, I should mention. We don't have contamination. Now I need to show you some data here. Obviously, we have some results here. Test tube data for pure and real genomic nucleic acids. There are complexities. Complexity is that genomic nucleic acids tend to be long. Not only long, they are like macaroni, they twist. And if I, by mistake, are targeting a sequence that is sort of like scrunched up in the middle, then we are not going to be able to detect it. We need to do something. So this is why testing genomic targets is really, really important. But we have been able to achieve 1 copy per microliter without amplification and in a single pot reaction at room temperature. So this is all published data. We played with the scheme. So we put some fluorophores on the circles as well to make what is we called a SER reporter. That works exactly like the previous one, and it is very much like a standard CRISPR sensor. You just don't give this fluorescent reporters like normal linear reporters, just circles, and it just keeps going and going and going. What is really interesting is that it is possible also to detect mutations. So we've done some proof of concept study with a specific cancer mutation, which was in colorectal cancer mouse. The signal was not particularly strong, but strong enough to be able to detect those mutations. I can't really tell you what the concentration was, but we estimate that the concentration was on a level of 100 attomolars. And the interesting thing is that one can make test strips with it. To make test strips with this technology requires a modification. And the modification that we have implemented in our published paper was this, that the circle was provided with two different molecules. We put a FAM fluorophore on one end and the biotin on the other end using a small single-stranded tail. So the Cas protein can cleave here to make this structure straight, and it also can cleave this to get rid of biotin. And so the process works in exactly the same fashion. The second big catalytic loop is activated and just breeds and breeds and breeds this fluorescent structure. And then they can be detected on a test strip. And the test strip works like this. This is the zoomed in thing that is inside of your plastic rat. And it has inside is a paper strip, which has different regions. This is the test line and control line where different proteins are immobilized. And here is a region with gold nanoparticles and anti-FAM antibody. When we run a sample from here to there, the control line just basically gives us a check that everything is working normally. And here we have the test line where in the colorectal cancer mouse model we had a stronger signal here. Regrettably, there is some background in the normal, but the signal is high enough to be able to detect both by naked eye, but also by the reader. We can quantify how much that is, and it is about 75 copies per microliter in blood plasma. This was a dilute plasma. So this is my last slide, and I understand that, mechanism is mechanism, but I wanted to really share it because it is probably the first time where we have a mechanism that can really be compared to the mechanism of LAMP and PCR. And just a few snapshots of what the advantages are. We inherit all advantages from CRISPR sensors. So high specificity, single nucleotide polymorphism detectable, high sensitivity, 1 copy per microliter. We hope we can reach zeptomolar, but we are not there yet. It is pretty rapid, and I have my team here in the room. We published 15 minutes. But I have been showing people some preliminary results, which were a couple of minutes. So definitely there is speed in those enzymes. No contamination, no need for closed reaction systems. This is such a relief. We also work with LAMP, which is, we are petrified each time we open the tube. It is feasible with freeze-dry reagents. We now have some lyophilized circles, so we know they survive. It's not really nothing novel, but you need to demonstrate this. That means there will be no need for cool change. Super easy to change sequences, but it's also a limitation. So because we work on 20 mers or thereabouts, if somebody is looking for more complex things in genes, such as aneuploidy, that's not it, right? There is only a limitation. But for infectious diseases, that's probably okay enough. We might be able in the future to look into antibiotic resistance, but that's another story. Sensors can be combined together in an OR way and user-friendly. A lot of interesting technologies can be aligned with this. So this is what we are doing, and our test strips use this. We don't make test strips. There is a company called Miltenyi Biotech offers kits that can be used to run those assays on. So that is really making everything very simple. We also work, typically our test strips are read on an instrument like this. This is an Australian company. But I am going to try to read this for you on a mobile phone as well, because this is the test strip readout is achievable on a mobile phone. This one is good because the data can be sent to the cloud in infectious disease control and public health. You want to have data in the cloud. And there are also big machines. We are working with the makers of those machines, Avicenna Systems, that can offer a whole lot of tests. Now, this machine is really big, 100,000 tests, 10 times faster than PCR. So we are working with them to give them the chemical engine of what the machine will be able to do. I think this is my last slide. Sensor platforms, I just leave this as a decoration. I don't know how we are looking for time, but I think we have 20 more minutes for discussion. So I had given you a snapshot of what the technology could potentially do in terms of technicalities. I didn't mention RNA detection at all because that would take us into the area of slightly more complexity. But I can reassure you that a lot of this can be repeated for RNA. The problem in the area of infectious disease is that pretty much every pathogen is bad, right? Some of them are worse than others. So I would be really, in theory, we can do anything we like, but in practice, no, because there is only one life, right? So we need to be really, really selective and help the infectious disease community to address diseases for which there is a market, there is a proper market. Now, the reason being is that this technology, whatever we think about the science, it can only be helpful for people if there is a proper commercialization pipeline. And it requires knowledge of market. It requires economic gradients to work as well. So I'm hoping that in discussions with you, we will be able to not only delve into the technicalities of infectious disease. We have spoken already, you know, that we are really not experts in infectious disease, but we are also not experts in commercial potential. And I would love to be able to discuss those two things with you. Thank you.
Well, thanks, Ewa, for a great talk. And I think you went through it really systematically. I've seen it before, so I guess I had an advantage in terms of understanding, but I think it was really well put together. If people have questions, could they go to the microphone so that the people on line can hear? And if people are online, could they enter their questions in the Q&A box? David, you can start.
Hi, Eva. Thanks so much. Very, very clear presentation for the audience. It's all about the target, as you were saying. And I'm not sure if we've had this conversation before about working with formalin fixed targets and whether it would be, if you've ever tried looking at trying to amplify from those, because that would be obviously a good resource to do testing in all those histopathological samples, just because the effect of formalin on the nucleotide structure based on shift bases and that kind of thing would be interesting to delve into.
Yeah. Thanks, David, for this question. The answer is obviously we didn't. I can't speculate what is going to happen. And the reason why I can't speculate is that the CRISPR reaction has specific conditions. There are specific buffers, specific level of this salt, those salts, magnesium ion, this and this and that, quite complex. And we are unable to predict what is going to happen if we drip feed some other compounds. Okay. It's unpredictable, but easily testable. In a formalin fixed sample, we are already talking about a laboratory experiment. So we are not in the field, we are in a laboratory. I can take your formalin fixed sample and do DNA extraction. That probably is going to give me a little bit of formalin or whatever are those evil molecules, but a little bit of adverse molecules is not such a problem. We know that there is a problem if we try to mix two different things. So we tried to mix LAMP reaction with its LAMP buffers with a CRISPR reaction. It took us how many? 6 months to finally zoom in at the combination that works. So we know it is able to be overcome. But the same consideration would not apply to trace components. So for comparison, we tried to run this reaction with a lysis buffer. Lysis buffer is pretty bad, but it was diluted and it wasn't a lot of it and it has worked. So I am optimistic we could try. We also didn't try dry blood stains. We would love to do that. That is such a good suggestion. You made it before.
Anya?
Thanks, Eva. It was, again, a fantastic talk, breaking it down for many of us in the audience. My question is sort of an extension from David's actually, which is about sample types. So formalin fixed is one type. Have you had experience in different understanding with the different infectious diseases, all the sample types that might come your way, swabs, urine, saliva?
We've done saliva research. We actually published it here. There are different compounds and enzymes that you can add to saliva. The first primary trick is to dilute a little bit. We have sensitivity. We can dilute by a factor of 10. That is frequently enough. Then there is proteinase K. We frequently like to, our biggest enemy are native nucleases. So we can sort of like suppress them using proteinase K. And my team has recently uncovered something else which I approved. There are some other similar compounds that are, you know, some proprietary compounds. What else have we tried? We tried blood plasma that works with proteinase K. We worked with plant extracts, but I think from memory they were already extracted, right? So that's not really applicable. Sample preparation is, of course, something that we need to be really respectful. I can't really comment on urine. Urine is probably, you know, has salts in it. That would be a challenge, but the system can tolerate salts, so it's a trajectory, right? If somebody tells me what we need to try, we will try.
Yeah, lots of things.
Lots of things are doable.
If it's okay to ask.
Lysis is obviously in the picture. You have to do lysis in many cases, but I would like to actually contest that a little bit. So our experience with COVID, we detected COVID, we published on COVID. We have COVID in the in the lab, that those COVID samples, these are not real COVID samples, they are reference materials, so inactivated COVID. And there was an instruction to say that, oh, inactivated COVID, you need to do this and that to release the RNA. No. Whether we release or if we don't release, the answer is the exact same. So it degrades, but the RNA stays in those materials. So there is natural degradation occurring that makes me think that for RNA viruses, maybe we don't need to do anything. Maybe we just need to wait a little bit so that things get aged. Now, for bacteria, the same, like bacteria will die and they will lyse. And if they will lyse, they will release DNA. That DNA stays because it's a robust molecule. So in my opinion, a good step is just to see if it's already there. And if it isn't, then yeah, you apply some methodologies.
So it was around, there's two aspects to this. One was with regards to the specificity of it. And you're speaking to the fact that we can do SNP detection. So clearly that's the antibiotic resistance is something that we'd all be very excited to explore with you. Related to that specificity is the opposite with regards to acknowledging a range of different technologies that also might have sort of that explosive effect. If you say that sort of rolling circle, that self-amplification cycle on top of the detection, if it is not 100% specific, the risk of false positives is also high then. So if it goes wrong, it goes really wrong. Do you know what I mean? So to counter that, reduce the risk of false specificity, is it possible in your system to have, for example, the need for 2 targets that then increase that requirement for multiple targets to be perfectly matched to get a signal like 1, 2, 3 equals yes?
So I can't comment on false positives yet. We have done some work on false positives. It wasn't on large enough samples and it was in lab situations. The results were good, but lab results that are good are not really, indicative of what is going to happen. Putting together reference tests makes a lot of sense. It would, at this point, it would have to be probably in separate wells if you are doing multi-well detection or on separate strips.
Okay, cool. Anyway, just something for us to consider as we...
No. I mean, I absolutely understand the requirement for different controls. It really needs much more controls than a normal person would think. There is lots to explore,
So just online, there was another question about extraction, but I think you've probably addressed that to some extent.
So for extraction, in my opinion, we are willing to undertake measurements without extraction. I think that should be tried first. And then if that doesn't work, then extraction.
Hi, Tyson here from the HIV and Epidemiology and Prevention Program here at the Kirby. Thank you very much for your presentation again. My question's around implementation of your technology, which is probably jumping a little bit further ahead than where we're at the moment. But you did liken that this technology could be seen maybe in rats in the future. What's your take on the user friendliness and maybe even getting to a point where we can implement these in self-tests like we have seen with the COVID rats, and then also cost effectiveness to ensure that it's actually accessible for community as well? So I was just curious. Thank you.
So it's really very convenient that this technology requires small volumes of material. The smallest test tube that we tested were 5 microliters. And I think our group has done some estimates on how much the reagents cost if they were purchased like an academic group would purchase. And we ended up with numbers well under $10 per test. The user friendliness. Now, we have made test strips here, some of which are shown here, and there are some newer test strips. We are working towards test strip for wider commercial distribution as well. The question is what type of sample will be in those test strips. So probably our current thinking is that saliva, because that's important for some diseases. But in the future, I can easily see fresh blood, the pinprick blood. And the reason being is that I have shown you what is going on behind the plastic on a rat test. There are pads, and then there is like a membrane. It's really like a paper filter. There is a very strong chance that red blood cells stay on this filter, and what flows is plasma. That's all you need, right? You don't want maybe red blood cells to be floating around or any other blood cells, but the rest of it is fine. So I can't answer, but we'll definitely try to work with fresh bloods as soon as we overcome the main problems. And the problems at this point are obviously scaling up. That's a conventional problem in all of this type technologies. So we are actively trying to overcome this barrier.
Hi, thank you for that really insightful talk. My question is whether your team has looked at modifying the Cas12a to be in combination with Cas13 to detect both DNA and RNA in the same sample. So for us, it would be like co-infection setting would be really important.
Yeah, absolutely. We have, and I didn't mention any of this, there are benefits of working with Cas13. There are some orthologs that are so amazing. The beauty of Cas13 is much faster cleavage. That can potentially lead to really those super fast assays. I mentioned that we have achieved like 1 attomolar 1 minute, right? That's completely crazy. It's scientifically, you are very excited when you see that, but in practical terms, that's not very exciting because you have to have a technology that is used by people in the field. They need to be able to stop this reaction because it is showing at 1 minute, but in 5 minutes, everything is gone, right? So this is the flip side of being fast. But yes, we absolutely can detect RNA, yeah. There is a number of proteins, Cas14, for example.
So just to continue on the question that Tanya started asking, and again, there's a question online from Marie Lundgren about how you would go about multiplexing this. And she's in particular is asking, can you use the cleavable probe to enhance the multiplexing?
So this is actually a weakness of Cas proteins. The specificity with respect to cleavage is not super amazing. So I have shown you a short region where we are wanting to do the cleavage, and there is some preference for some nucleotides, but it wouldn't be multiplexing for a lot of different colors. What I think is going to happen is that in the short term, we will be developing test strips with multiple lines. The machines I've been showing you, especially the Avicenna machine, is completely able to multiplex in different wells. And from those end user point of view, that's fine. The sample goes into the machine, an untrained person slides or keeps giving the test tubes with saliva into the machine, and then machine does everything, and then you just have an answer. But multiplexing in a sense that in one test tube, you've got different colors emerging and up to 10 colors, that's not going to happen soon.
So Ava asked us earlier what we thought were potential targets. So does anyone in the audience want to volunteer preferred targets? I mean, Ava and I have talked about this quite a bit, but
just to add to the conversation, we'll be chatting about sexually transmitted infections such as Neisseria gonorrhea, which has those high need, high burden, and a need for the mutation that's SNP detection as well with regards to antibiotic resistance. So it's a world of opportunity, I think.
yeah.
Thanks for an amazing presentation. My name's Andrew Grulich. I work in HIV prevention and I'm a public health person, so I don't understand the methods, but I'm excited by the possibility. So I'm just interested, a lot of DNA detection is a fairly mature science already, and we're very familiar with a lot of techniques that use that. So what role do you think this has in a field which is fairly rich already with DNA? How are you going to sell it? Is it increased sensitivity? Is it ease of use? Or is is the absence of the need for amplification what it's all about?
I don't have PowerPoint, but this was covered at the beginning. So a good scenario would be a scenario where you are testing environmental DNA. So in environmental DNA situation, somebody goes out in the field and here is a river and we wanted to look for, I don't know, koalas. So we take a sample from this river and obviously do we want to really take the sample and send it to the pathology laboratory here and you are somewhere in the interior.
Yeah, but my point is about that, you know, for many pathogens, we already have rapid diagnostics that we use in the field, that we've already got them. And I'm wondering whether you feel for those conditions where we already have rapid diagnostics, is this going to overtake them because it's more sensitive or because it's easier or cheaper in the field? Or
I don't think it is more sensitive. I wouldn't be able to tell you whether it will be more sensitive. Currently, it isn't. Cost per assay can be lower. And it would be probably a unique opportunity to test outside of pathology laboratory. When we are thinking about the disease, what sort of the picture that we have is that here is a person, they are already very sick, they are in the hospital, here is the hospital pathology over there, no problem, we are going to test them. In circumstances such as this, maybe cost is not a factor and the logistics is not a factor, but there is plenty of other situations when it is a factor. For example, an example from Tony, I'm going to quote Tony, who told me that maternal HIV transmission in the Pacific is a significant problem. And in order to detect the fact that the mother or future mother has an infection, the time from taking the sample to the answer can be many months. And by that time, the baby is born and then it is too late. So there is, and it is not just, I believe it's not just here, but also in Australia, there are circumstances where pathology time is a significant factor.
And some researchers in the Kirby, in fact, this is Tanya's group, are already doing this with PCR-based machines, but it takes 60 minutes and and it's complex. So I suppose the advantage is that if you can get this on a strip, it's going to be much better than that.
It's not assay time. You have to have a machine at the location.
Yeah, yes, that too.
Right? So the machine also needs to be simple enough to be operated by untrained personnel. And I understand that those machines have been placed in remote locations by the virtue of grant funding that you had. And if there is no grant funding, then there is a problem.
Thank you.
So I think the penetration of the technology depends really on how cheap it is. And how user-friendly it is. So hopefully this is going to be more user-friendly, but you know, time will tell. It took PCR many, many years to come out from the scientific laboratory and become, you know, the everyday technology. And even now it is not everyday technology. It is still in professional labs.
Thank you.
So we're at time. I think that's been a really excellent presentation about some really exciting technology. I think this is, you know, something that we should be working together on. I hope that this is the start of a long-term relationship. So thanks everybody. Thanks Eva for a terrific presentation, and thanks everybody for coming along in the very active discussion.